Graham and his staff. Graham is going to do something that we think is really important as you prepare for your lectures or whatever introductory materials you're going to do with your students, and that is he's going to give you an overview of phage biology. He's also going to give you time to ask questions. So Graham is an HHMI professor. He's also a full professor, and he was the chair. Is that the right verb? Was? Okay, he's chair of biology at the University of Pittsburgh, and of course in Pittsburgh, Pennsylvania. It gives me great pleasure to introduce Graham Hatfull.
Well, thank you very much, Tawanda. I'm not sure I've ever been a lead anything, let alone a lead scientist. But there can be perhaps nothing as much fun as having just such a large group of folks who are sharing in the joys of bacteriophage biology. It's something that I've been growing to become quite fascinated with over the past 20 years or so. And so having all of you involved in this activity and in this workshop this week is really super. So I have, we have about almost 2 hours this morning, and so I'm going to try to talk to you about bacteriophage biology, really beginning from a more basic and historical perspective, and then lead up towards where I believe we are today and the things that we may do or want to do to understand the population of bacteriophages. I'm going to split the talk into approximately 2 halves. The first half will be more talking about some historical aspects, thinking about lifestyles, morphotypes, etc. And then I'll have a brief summary, and at that point we can take a breath and I'll address any questions at that point. And then I'll move on to the second part, which is primarily going to be thinking about what the bacteriophage genomes look like, what the genes look like, and how we can think about them and understand them. Okay, so we can start, I think, by just reflecting back to the latter part of the 19th century when the great microbiologists, Koch, Pasteur, and many others, really made some stunning breakthroughs in understanding the microbial world, the prokaryotes, bacteria, germs and the causes of infection and human disease. And much of those discoveries were powered by the invention of the light microscope, the ability to be able to see entities that are so small that they're hard to see by the naked eye. And I think that that clearly was a revolutionary time in our understanding of microbiology, and many of the mysteries of the microbial world were settled and resolved and understood, I think, in a quite definite and finite way. And so it was something of an inconvenience about 30 years after that, that it was became clear, largely through the work of these two guys, Felix Durell and Frederick Twort, that independently came upon the discovery of what they believed was some additional biological entities in the microbial world, but which seemed to have very different properties from the prokaryotes and the bacteria that had been described previously. And I'm sure there's many versions of this story, but the tale goes that for Felix Durell, that he was treating a patient, a soldier, I believe, who had an infection, and he was taking samples from this patient, and he was growing the bacteria. And there was one day when he came back and he took a sample, and he was mixing it. So the sample was from the patient. He was mixing it with the bacteria that he'd been growing, and he observed that that the culture, which was turbid with bacteria after he had infected it, went completely clear. The bacteria had been magically killed or cleared. They were not visible anymore. And the story goes is that Durrell figured that if whatever he had added to his culture of bacteria and had killed them was present in the patient, then the patient should have benefited from the very same effect as he'd seen in the test tube, rushed over to the hospital the next day and discovered that the soldier was sitting up bright and cheerful, the signs of infection had been cleared, and Durrell was actually inspired from that point onward to spend virtually the rest of his life trying to see how bacteriophages, which he had discovered, could be used for therapeutic purposes.